Tuesday we'll do another full review session where you get to review with your friends and peers, and I'll be here to help you with that. And then Thursday you have your second exam. It's going to be just like the first one. It's going to be about half multiple choice, half coordinated in there. Questions? Any logistical questions? Some of you had some conceptual questions, and I asked those people to wait for that to the end of today's lecture, but I absolutely want to go over it. It's a topic that almost everybody wants to hear 2 or 3 times, so I think if you'll wait till the end of the lecture, I'll review that. I think it's about the lack opera. I'll review that, and pretty much everyone in the room will benefit from hearing it at least one more time, and I'm happy to do that. All right, any questions, other questions today? What we talked about on Tuesday was microbial evolution. Before that, we talked about lateral gene transfer, horizontal gene transfer. Then we got into evolution and a little bit about taxonomy. What I'm going to go through today is getting into some nitty-gritty, hopefully not too dry, description of the diversity of so can I answer any past questions before I get started? Alright, so what we're going to do today, many of you probably read the paper for today, and it talked a lot about why we care about whether or not microbes are diverse in terms of function. And we'll get to that at the end of this lecture. But what I want to go through today fairly quickly, and I'll let you know when you need to focus more or less. What we're going to go through today fairly quickly is just a good overview of, and it's shameful that I'm fitting all of this into one lecture, a good overview of the diversity of microorganisms in terms of their taxonomy. And the differences, and I'm going to bring up some organisms that you've heard of, organisms that are very relevant to your life, and some that you haven't heard of that are relevant to your life when you know about it. All right, so this is a giant phylogenetic tree that was cut out of your book, copied from your book, and it spans across 2 pages within your book. And what it does, it breaks up, remember what we have with respect to, this is just the archaea and the bacteria. Remember you have the 3 domains of life. You have the bacteria, the archaea, and the eukarya. I actually had some interesting conversations with my colleagues in the last few days. Apparently you guys were taught the kingdom system. So raise your hand if you know the kingdoms. The kingdoms is in Monera, Protista, Plantae, Animal. Wow, okay. I pretty much thought that was obsolete, but I live in a very microbially centric research world. Those kingdoms fit within the 3 domains. For the purposes of this course, please learn it as the 3 domains. I personally feel that's more accurate. I didn't realize it was still up for debate. I thought that was done and put to rest now that we have DNA technology. But some people apparently still use that at the earlier level. The 3 domains, let me be clear though, the 3 domains encompass that. Okay? So, the kingdom Monera, which is just hilarious to even say, fits within the archaea and the bacteria. So bacteria and the archaea actually encompass Monera or within that. Part of why I think it's so scandalously inaccurate. And then obviously the Protista are eukaryotes, animals, plants, they're all eukaryotes. But don't let that confuse you. And if it does confuse you, please raise your hand and I'll clarify further. But the 3 domains of life is the big way to look at it, the big molecular way to look at it based on understandings of DNA and sequences. All right, so what we have here in this slide and the next slide is really just a quick look at the archaea, and then we're going to break down the phylogeny of the bacteria. So I would argue the way your book presents it, it just very briefly presents the archaea, and I'm only going to tell you just a tiny bit about the archaea. And we're going to spend more time on the bacteria. And one of the reasons for that is that we just know a lot less. It is definitely not that they're less diverse, less important, less abundant, less ubiquitous on this planet. It really is just that we know a lot less, and we spend a lot less time writing and thinking about it and then putting that into textbooks. But they're absolutely just as diverse. All right, so the archaea can be broken up into the Euryarchaeota and the Crenarchaeota. And some of the important take-homes, I'm going to give you some concrete examples of this in just a little bit in a couple slides, but some of the important take-homes from the archaea that you guys already know and you've actually given to me on exams is they tend to live in extreme environments. Not exclusively. Some of them live in fairly moderate environments. You can certainly find them in moderate environments. But they tend to dominate some really interesting metabolic functions in very extreme environments where you maybe have extremely low pH or very high temperature. And you remember archaea have different cell walls. Their cell walls lack peptidoglycan. Their ribosomes are a little different. Etc. And we'll talk a little bit about some examples of archaea in just a minute. All right, now we're branching off into the bacteria. And within the bacteria, we have the thermophiles. Okay? And within the thermophiles, the important things that you guys care about, and many people in this room care about, is we originally got TAP from a thermophile. What is TAP? Who remembers that? Maybe I brought it up on the last lecture, a couple lectures ago. It's for PCR, exactly. It's the enzyme we use to carry out PCR. And the reason that enzyme is handy and the reason it was isolated from a thermophile, let me see if one of you can tell me. Some of you that have done the PCR, why do you think? Very heat stable. Is that where you were? Yeah, it needs to be extremely heat stable. So not only, yes, to some extent they've engineered it that way more recently, but originally it came from a thermophile. All right, and then we're going to break it up into the gram-positives and the gram-negatives. So it is not trivial, it's not meaningless that we've made you learn gram states in lab. When you get to the last few labs in the laboratory section of this course, you're going to get to identify your unknown, and hopefully we're going to have at least a couple of you be successful through biochemical means, as well as doing a sequence of your PCR product, your 16S PCR product. And first thing you're going to do before you do those biochemical tests is you're going to break it up into either the gram-positives or the gram-negatives. So you're going to do a gram stain on your unknown and see where it falls in that phylogeny. It's the first way to break them down. All right, so within the gram-positives, you have low GC gram-positives. Those are the firmicutes. And you have high GC gram-positives. And those are the actinobacteria. Your intestines are almost completely dominated by firmicutes. All right, you got plenty of enterobacteria in here as well, but one of the biggest things in terms of absolute numbers are firmicutes. All right, actinobacteria that are high GC bacteria, they live in soil. Those are your actinomycetes. So they're bacteria that sometimes act like fungi, look a little like fungi. And they live in soils, you find them rampantly in compost heaps, etc. So someone tell me the significance of the fact that some organisms are low GC versus high GC. What do we mean by that? Why are they classified in that way? Take a guess. I don't expect you to know, just take a guess.
In the DNA structure, they have less GC combinations, right, and they have more ATs.
Absolutely, right there. They have less GC combinations and more AT combinations. And what does that mean for their DNA? One. She's on the right track. There's another way to put it. I think it's less hydrogen. Okay, we'll say the same thing right there. You're really close, but what does that mean for the environment they can live in? Victoria, are we going to make a guess?
High GC, the DNA molecules rich in GC can resist like changes in temperature more.
Yes, absolutely. For all the reasons that these people are saying, absolutely. So at a high GC content, they're going to be more tolerant of high temperatures. Right? Hence composting. We have to spend a lot of time composting. Compost gets really, really hot, and actinobacteria thrive in there. That's just kind of a gross generalization, but it's one way to think about it and try to remember. All right, so let's move on to some people. Okay, so on the other side of this is in your book spanning a couple pages. All right, we also have, now we're on the side of the gram-negatives. And we have the spirochetes, which their picture, comes right there. Spirochetes are pulled from Lyme disease, Borrelia burgdorferi, and spirochetes. We'll tell you a little bit about these a little bit. A lot of those spiral-shaped organisms that do funky things and mess up your head. All right, have photosynthetic gram-negatives, and then we have the proteobacteria. Photosynthetic bacteria are obviously things you've heard a lot about, like the cyanobacteria. You spend a lot of time looking at those guys in the lab and the microscope. And the proteobacteria, this is the biggest, most well-known group. All right? And one of the reasons for that is that gamma proteobacteria are all the things that are human commensals in pathogen. Not all of them, but a lot of them. I also told you the firmicutes are human commensals in pathogen. So a lot of the things that we think of that live in our intestines, like Salmonella and E. coli, Klebsiella, the Enterobacter, they're all in gamma proteobacteria. Alright. And then you have some funny things with the Delta proteobacteria. And it has organisms like Bella vibrio, which I'll tell you about, which is a predator that does some pretty neat stuff. Okay, so let's go into, I don't want to spend too much time. Let's start breaking these groups down. So we'll go through the archaea first. Okay? Just very roughly, let's just talk about the phylum Crenarchaeota. Those are most of those extreme thermophiles. Think of hot springs out in the middle of summer. Many of them are strict anaerobes. Some of them can even live at extremely low pH. And since I just told you they're anaerobes, and I'm telling you they're sulfur dependent, many of them are sulfur dependent anaerobes, then what do you think I mean by that with respect to, I'll give you a hint, with respect to their respiration? Anaerobes sulfur dependent.
They respire sulfur?
They respire sulfur in what form?
Gaseous sulfur?
Gaseous sulfur? What, and what biochemically?
SO2?
H2S, and was that gonna be with a, is that gonna go into the cell or go out of the cell?
They dump their electrons
Do they dump their electrons on H2S?
on sulfur.
No, sulfur. What kind of sulfur? What kind of sulfur do they dump their electrons on? Hmm? Oh, you guys are doing, there's people in this room that have a test on Tuesday, and I know they should, and they're laughing and hiding behind a coffee. Alright. They're dumping their electrons on what, Anthony?
Sulfate.
Sulfate,
And
beautiful.
releasing
Knew you guys
H2S.
knew it in your quiet. Okay, and what?
And then releasing H2S.
And they're releasing H2S, absolutely. So in an anaerobic environment, they can use alternate terminal electron acceptors, something besides oxygen. And you guys were all kind of getting mad. Decided to focus on the other test soon. So they're dumping their electrons on sulfate. And in the process, reducing it down to hydrogen sulfide. So hydrogen sulfide is what they're respiring now. Huh? Alright, so they can be organotrophic, even in that anaerobic environment, they're just dumping their electrons on something else, frequently sulfate, not exclusively. Or lithotrophs. Okay, so they can also get those electrons and energy from something that's not organic. And in that case, they would be autotrophic, getting their carbon from where? CO2, good. And it's a huge group. It's a big, big diverse group. Okay. Again, thermophiles up to 80 degrees Celsius. They can live in fairly low pH. They can be lithotrophic on sulfur. In some cases even be aerobic using oxygen or iron. And they can also be organotrophic using sugars and amino acids. Okay, so let's go to those gram-negatives now, the non-proteobacteria gram-negatives. It's going to be things like photosynthetic bacteria, the cyanobacteria. All right, and this is such a freakishly diverse group. You think of cyanobacteria as just little floating plants, and yes, you're absolutely right, autotrophic, taking in carbon dioxide. But the difference from the cyanos than just your average plant or your average grass sitting out in the lawn right here is that they use a bunch of different pigments. They don't all just use chlorophyll the way your average plant uses chlorophyll. They use a lot of different pigments, which allows them to optimize many different environmental niche. They can also, strangely enough, be heterotrophic. So they can be heterotrophic, but they get their energy from light. So they can be taking in organic carbon, but then also getting energy from light, living this kind of a half photosynthetic, half, half photolite, half heterotroph life, almost, kind of hard to describe and define. Okay, so gram-negative. Many of them are obligate photolithoautotrophs, but not all. Okay, some of them can be chemoheterotrophs. They can be unicellular, they can be colonial. Many of them that you looked at in the lab are colonial, which is good. It makes it easier for you guys to look at them. They can be incredibly pretty. These are the ones that, going back to the idea of the kingdom designation, you may have even heard of cyanos as being the blue-green algae. How many of you heard of the blue-green algae? That's familiar. Cool. These are the blue-green algae. And it's because of those differences in the pigments that they have. They don't just use chlorophyll. They use a lot of different pigments, making them really beautiful colors. They're taking over my fish tank at home right now, and it's quite pretty. It's not annoying. It's this carpet of kind of blue-green all over the fish tank. Beautiful and a pain to clean. Okay, cyanobacteria, they can grow anywhere. My fish tank, they can grow in really hot environments. Remember, that maybe was in a different class. I've showed pictures before of some of the pools and ponds in Yellowstone Park that have these amazing different colors. A lot of it has to do with cyanos that grow optimally at different temperatures and have different photosynthetic pigments. They're frequently the primary colonizers in an environment, frequently the first to arrive and take over, and capitalizing on nutrient-rich pools. And a lot of cyanos will produce toxic secondary metabolites. So what I mean by that is some of their own metabolism produces a chemical that can be harmful to other organisms. Part of why a cyanobacterial bloom can cause all sorts of nasty pollution events, kill off fish, things like that, kill off other organisms. Not just because they're depleting oxygen, but also they're just flat out producing toxins. Big problem in things like golf course ponds where they're put a lot of nutrients on the grass and it runs off into the pond. And I'll give you more about that in a different perspective in a little while. Okay, so again, this is just reiterating. We have a lot of different kinds of photosynthetic bacteria, many different functional groups. And what I mean by functional groups is how are they functioning in their environment. And this is just looking at kind of their pigment profile, where some of them have most of their pigments down here in the lower wavelengths, some of them can actually absorb out there in the red area, etc. The take-home message from this figure is that some of these organisms, this is just chlorophyll, microvilli proteins, carotenoids. Some of them can absorb in a lot of different wavelengths. Some of them absorb very far apart and in different wavelengths from these guys. Okay? They're not all the same. They do not all overlap the same in the wavelengths that they absorb. What this allows for is them to live in different parts of the water column. Light doesn't move evenly through water. All the wavelengths do not move evenly through water. Some organisms can live at deeper depths. Some of them can occupy shallower depths, depending on the photopigments they have that are most optimized to whatever wavelength, allowing them to occupy different niche. And those of you that remember your ecology, if they're occupying different niche, then what does that mean for them? If I can occupy a different niche than somebody else, what does that mean in terms of resource use? Why is that a benefit?
More resources.
Beautiful, there's no competition, it reduces competition. And just a little bit about this. Really important symbiotic relationships. A lot of them are associated with the corals, we'll talk about later on in the semester. Huge effect on the global carbon cycle. When we think of, you think of things like the rainforest is really important carbon sink globally as we try to reduce atmospheric emissions and try to reduce carbon dioxide and methane getting out into the atmosphere. Yes, it's absolutely true. The best carbon sinks are things like phytoplankton and cyanobacteria because they're photosynthesizing so fast and turning over their, think how fast they divide as opposed to how fast a giant tree divides. Really, really quickly. So they're taking carbon dioxide, fixing it into sugars really fast, and pulling it out of the atmosphere at a much faster rate. Even though they're so microscopic and tiny, their rate of turnover is so much faster than a big plant or something that it's a very important part of the global carbon cycle. tiny, tiny percentage of them, but they're turning over so fast, using so rapidly cycling carbon out of the atmosphere and into some sort of biomass, organism biomass. They're really important for global carbon fixation. All right, so let's move on to spirochetes. Spirochetes are neat. Again, gram-negative, chemoheterotrophic. Again, organisms like Lyme disease, which we're going to spend some time talking about in the disease portion of this course because it's so relevant in this geographic area. Other things you've heard of like syphilis. Okay. They have interesting relationships too. Spirochetes play a very important role in the guts of termites. And we're going to spend a little bit of time talking about that in the third portion of this course. Anyway, also Lyme disease again, carried on ticks. We'll spend some more time talking about that with respect to the disease portion of the course. And here's just a picture of some spirochetes under the microscope. They're incredibly pretty. And they can be incredibly huge relative to other bacteria. You guys all know now what it's like to look at a bacteria under the microscope and how tiny they are. Even at 1000x, you look down and you just see a dot. It's kind of anticlimactic. Spirochetes sometimes, though, are really, really impressive. They can get big. All right. Spirochetes, that was just such a short introduction. We will spend some more time on those later on in this semester. They do a lot of neat things. All right, so the proteobacteria. I need to spend some time on the proteobacteria because, as I said, it's such a big, important group, and it's so much of our own human commensal group, proteobacteria. And then I want to just spend a little bit of time talking about the Vibrio because they're fun and interesting. And it's a good way to get you guys introduced to the importance of bacteria and in the environment and habitat and the life history pattern that you probably never even considered. All right, so incredibly important in your life that you've never thought of is the organism Rhizobia. Rhizobia are in the alpha proteobacteria. They are gram-negative. And they grow symbiotically with nitrogen-fixing plants. But the plants aren't doing the nitrogen fixing, the Rhizobia is doing the nitrogen fixing. All right? And we'll go into this again. This is another idea that we'll go into a little bit more detail in the third part of this course. But leguminous plants are very, very special. And one of the reasons they get used heavily in agriculture is because they harbor these symbiotic bacteria. Rhizobia is one of them, but not the only one. That takes atmospheric nitrogen into gas out of the atmosphere and metabolically converts that into organic nitrogen. This is a really, really big deal. We put organic nitrogen as fertilizer under crops in great quantities so that we can grow more crops, particularly things like corn, clog up a bunch of nitrogen. All right? Leguminous plants with their rhizobia do that on their own. You don't really need to fertilize peanut crops, bean crops, soybeans, clover, alfalfa. Those are all legumes. And they harbor this bacteria. So as much as you take it for granted, this bacteria is the reason that we have enough food on this planet. It gets nitrogen out of the atmosphere to turn into proteins in plants that can later support consumers. So not just the plants and the grains that we eat, peanuts, soybeans, etc., but also the things that eat those things, cows, sheep, chickens. It's the base of a lot of food. So this bacteria grows within the roots of the plant in these nodules. And you can actually see those nodules if you pull up the roots of, say, clover in your backyard. You can actually wash it off. You can see some of those nodules of those nitrogen-fixing bacteria. And so what they're doing, the process of converting atmospheric nitrogen into ammonia is an extremely metabolically expensive process. Greatly reducing that nitrogen to ammonia is very expensive, and so they get carbon from the plant in exchange. So the plant gives the bacteria sugars, and the bacteria give the plant nitrogen and all the soil around it. So other organisms benefit from the legume being present as well. All right, here's a cool example. Vegetoa. It's in the gamma proteobacteria. Microaerophilic. What does microaerophilic mean?
Allows the oxygen.
A little bit of oxygen. Yeah, it can handle a little bit of oxygen. The reason they like to live in a little bit of oxygen is they like to live in kind of a right at a threshold. So Beggiatoa, where they are, going through anaerobic sulfate metabolism, reducing sulfur down in the anaerobic sediments, but getting organic matter from above. So you have kind of a combined metabolism where they like to live where there's just a little bit of oxygen, and they're getting organic matter from above, and then reducing sulfate. One of the byproducts of that is elemental sulfur, and you see that as this white carpet out there on the bottom of the water column in the sediments. All right, so this is a fun one. Dellavibrio and the delta proteobacteria. Dellavibrio is this active, you can see it, you can watch it happen under a microscope. I've never been lucky enough to spot it, but I've had it. Where this little bacterium has a flagella and it's highly motile, and it leads a predatory lifestyle. So something that's been implicit this entire semester that we haven't really talked about that is how do bacteria forage and get their food? And you think of it as they're just these dumb little cells lying there floating, waiting for glucose concentrations to rise. It's pretty much true. Dellavibrio, though, is a predator. And it actually swims and attacks into a cell and will penetrate into a cell and then produce more Dellavibrio, parasitize that cell, and explode, almost like a phage. But you can watch it. Like I said, a buddy of mine has actually seen it happen. You can watch them dive and penetrate. I should look for a video. I'll try to do that. You can find it. I bet we could find a video on YouTube where you can actually see them penetrate down into the cell, and they'll charge at it and pound into it. So that sounds awfully directed and with premeditated thought for a bacteria to do, but they do. All right, so let's quickly go on to the gram-positives. The gram-positives, remember we have high GC and low GC gram-positives. All right?
Bless you.
In the low GC gram-positives, we have things like Clostridium. So you guys have spent a lot of time looking at Bacillus in the lab. Bacillus is also a low GC gram-positive. And Bacillus and Clostridium have an interesting thing in common. Who knows what it is? Yes, from this picture. Someone said it's spore production, yeah. They both produce spores into spores. Clostridium is highly anaerobic. And that's part of the difference in its lifestyle. Bacillus in many cases can be, but Clostridium is certainly going to almost always be anaerobic. Anyone know any of the cool diseases that Clostridium causes? Botulism. Cool. Absolutely. Your Botox that you're injecting into your lips is botulism toxin. What else? What's another one? Tetanus.
Yeah.
Good. Yeah. Tetanus is another one caused by Clostridium. So, all right. So these are anaerobic. The reason that you think about botulism is you've got a can of beans that's been sitting in the cabinet for 10 years and it's puffed up and it's all of a sudden producing a bunch of gas. It's because it had botulism in it. And it's sort of growing anaerobically in there and producing gas. So that's bad. Anyway, so botulism is a disease associated with rotten food and that kind of situation, an anaerobic case, but not exclusively. Tetanus, you think of rusty nails, that kind of thing. Anaerobic soils where you might get tetanus. All right, actinobacteria. So the actinobacteria, actinomycetes are a kind of actinobacteria. Do not mess you up and confuse you. They're members of the high GC gram-positives. And as I said, this is a ubiquitous group living out in the soil. Anytime you've had something like a nice hot compost pile, you're going to see tons of actinobacteria in there. And they grow in almost a filamentous fashion, partly why we call them mycetes. Mycetes is indicative of a fungi. And it's the word we use to describe fungi, so they borrowed that word to describe an actinobacterium that's growing in almost a fungal-like way. But one of the main reasons you care about the actinobacteria is almost a huge percentage of our antibiotics have come from the actinobacteria. Have been isolated from this group of organisms. Streptomyces is one. No, Streptomyces is the organism. Streptomycin. Am I saying that right? No, I can't think. Now that I've thought about it, it's like, I don't know. Anyway, you get my point. A lot of important antibiotics come from actinomyces and actinobacteria. And again, they live in soils, those kind of environments. environments that can potentially get extreme. Partly that explains their high GC content, but not exclusively. Yeah, as I said, they can grow in almost a very fungal-like fashion where they'll produce spores, and then they'll grow and produce chitin, and they're running like chitin in the same way that fungus does. Right. And they can do some interesting, they're very important for decomposition. They can break down things like lignin. Lignin is the hard polysaccharide that holds, makes plants rigid in terrestrial environments. So hugely important to decomposition. All right, I want to make sure I have enough time for everything today, especially because some people want to review some other things. I'm going to jump around just a tiny bit.